actually. Do we need to have a working camera for the final exam? No, it's really no different than you taking the test. I think somebody asked me if I could have it open until midnight that night, and I don't see why not. So I can set up that way too. But you'll still just have your allotted time for it. Okay, metabolism. I thought this was an interesting fact. Average person over 40 years will consume 6 tons of food. Wow, that's a lot of food. And 10,000 gallons of water. There are two types of metabolism. There is catabolism, which is taking larger molecules and breaking them into smaller molecules. This is what your body does when it's digesting foods. And this produces energy in the body. You also have the reverse of that, anabolism, which takes smaller molecules plus some energy and builds these larger molecules. So for example, we eat proteins. We uh, we go through catabolism to break those proteins down into amino acids. And then when our body needs to build protein, like you're weightlifting or something, and you're building muscle, it's going to take those amino acids. It's going to use some energy to do this, and it's going to reverse that and build the proteins out, which is just what we studied last week, how the body goes about building that protein in the ribosome. And so probably some of you have heard of anabolic steroids. This is what it's referring to. Anabolic steroids is where body people that are into bodybuilding will take them because it will build more muscle mass. It's also very unhealthy for you, so don't do it. But they will do this in order to build more proteins, therefore more muscle in their body. There are two types of metabolic pathways. There's a linear one where it's just A produces B, produces C, produces D. The one that we're going to deal with mostly are cyclic metabolic pathways, where you have A produces B, C, D, and then D produces A again. This is a very common pathway in the human body, or an animal body for that matter. There are two types of cells. There are prokaryotic cells, which do not have a nucleus at all. These are typically found in bacteria. So you see here is a picture of one. There's no nucleus inside of here. The cells that are in our bodies are eukaryotic cells, which do have a nucleus which contains DNA. So here we have our nucleus in the center here, this orange globe in here surrounded by a membrane, and then the outer contents of the cell. And we'll get into this more in a moment. Like right now. So main components of a cell, like I said, is probably review for a lot of you guys. You have cytoplasm, which is the water-based material that is between the nucleus and the outer cell membrane. So that's all of this stuff that's outside of this nucleus right here. Cytosol is the water-based fluid of the cytoplasm. So it's this light green shaded area in here is the cytosol. It's kind of like the solvent, if you will, that this stuff is in. An organelle is just simply a thing inside of a cell that carries out some kind of function. And the two main ones that we'll talk about are lysosomes and mitochondrions. Lysosomes are where you get hydrolytic enzymes for building cells and repairing cells or tearing down cells that have died. Some of lysosomes will hydrolyze proteins into the amino acids, breaking those down. And some will hydrolyze polysaccharides down into monosaccharides or single sugars. So that's kind of the place where that happens. And you can see here, it's rendered here as these red structures within here. And mitochondrion are these orange structures, which is where we're going to spend most of our time tonight. This is where most of the energy for a cell is generated. So mitochondria are the powerhouses of the body. And we also have ribosomes in here. This is those construction sites for proteins are also in there as well. And of course, you have an outer membrane of the cell, keeping the cell intact. That's just a bigger picture of it. So the mitochondria itself, there is an outer membrane that's exposed to the cytoplasm. This is made up of 50% lipids, 50% proteins, and it is permeable. So stuff can move back and forth between that outer membrane. The inner membrane is 20% lipids and about 80% proteins. And that's this curly inner membrane that you see here. And this is impermeable. This is the bilipid layer that you have to have the proteins in order to shunt stuff back and forth between these things. The matrix is inside of this inner membrane, so it's the insides of this sort of curly structure. And the inner membrane space is the area between the outer membrane and that inner membrane. And this becomes important because this is where the chemistry occurs, is across this inner membrane layer. The folds of this inner membrane are called cristae, and it is at these folds where we have an ATP synthase complex, and this is where ATP is formed. ATP is central to the production of energy in the body, as we will see. But you can see here, it shows like these little knobs on the inside wall of this and the matrix side of things. And this is where the body will take ADP, react with an inorganic phosphate, and create ATP. We'll talk about this more in a moment. I don't think we've talked about ADP and ATP yet, have we? Or was that in a previous chapter? Not sure. Any questions there at all? Is this mostly review for all you biologists out there? Yeah, I figured. Like I said, you probably know more than I do about this. So ATP is adenosine triphosphate. We see an example of it here. So this is that base adenine, and it attaches to a ribose, and this forms adenosine. So these two together makes adenosine. And then it can be monophosphate, diphosphate, or triphosphate, depending on how many phosphates are hanging off of this C5 carbon over here. And so down here you can see it kind of in a block structure. AMP is the abbreviation for adenosine monophosphate. You've only got one phosphate hanging off the ribose. Adenosine diphosphate has two, and triphosphate has three. So ATP and ADP, so having three or two, will readily hydrolyze, and they will produce basically they'll lose a phosphate off. So ATP can lose a water, react with water and lose a phosphate. ADP can react with water and lose a phosphate. So the ATP will become ADP because you've just cleaved off one. However, ATP could react with 2 moles of water and give you monophosphate as well, plus 2 phosphates off. So 2 of the phosphates can cleave off with this kind of a reaction. And ADP just loses 1 because that's really all it has. So basically you just get down to that monophosphate. This produces energy that gets used in cell processes. The reason why is that more energy is produced than what is consumed in this reaction. So you have this extra energy now that can be used for processes within the cell. These phosphate bonds here are strained and they will readily break. And that's why it doesn't take much energy to break them at all. And that's why we get a net positive gain in energy. There are some other triphosphate nucleotides. There is uridine triphosphate that's used in carbohydrate metabolism. We'll see this next week. Guanosine triphosphate, which is used with both proteins and carb metabolism. And cytidine triphosphate, which is used in lipid metabolism. An important component in these reactions in the cell are one of them is flavin adenine dinucleotide. This gets abbreviated FAD. I call it FAD. And what it looks like is like this. So you have your adenine base, you've got your ribose sugar here, and then you've got 2 phosphate groups. So this whole piece right here is ADP, adenosine diphosphate. But then you have a ribitol and a flavin added onto the end. And these two together make up a riboflavin, which is vitamin B2. As we're going to see, vitamin B plays a big role in a lot of these reactions in the cell. Sorry, trying to get my straw to go in. FAD is used in redox reactions in the metabolic process, as we will see momentarily. And again, I'm not going to ask you guys to memorize or draw this structure or anything like that, but knowing that FAD is involved in a redox reaction in the body is what's important to know. What happens is FAD gets reduced. So here is the active part of FAD, and we have here a nitrogen with a double bond and a nitrogen with a double bond. These will react with 2 protons and 2 electrons to break those double bonds and give us NHs here, NH bonds. And so FAD H2 is a reduced form of FAD because we've reduced it because we've added protons to it. And so in this process, we're consuming 2 free protons and 2 electrons in order to reduce the FAD to FADH2. FADH2 will quite often pick up these hydrogens by taking a saturated compound and pulling those hydrogens off and creating a double bond on that compound. And now those 2 hydrogens can react with the FAD and give you FADH2. Any questions there? Nicotinamide adenine dinucleotide, another mouthful, gets abbreviated as NAD+. And this is also used in redox reactions. It does have a positive charge because right here we have a nitrogen that's got 4 different bonds. It's got a double bond to this carbon, a single bond to this carbon, and also a single bond to this carbon. So remember, nitrogen only takes 3 bonds, so this must be in a positive state. So this is nicotinamide, which is a derivative of nicotinic acid we have over here, and it's connected to a ribose. It's connected to 2 phosphates, another ribose, and an adenine base. So it's basically, you've got ADP combined with another ribose and this nicotinamide in order to form NAD+. Niacin is vitamin B3, and this is a precursor to nicotinamide. So another vitamin B is central in these reactions in the body. Any questions there? Is this review for you guys, or was it more the biology stuff and now we're into chemistry stuff? And so this is new. That would be my, pardon me?
This is new for me.
Okay, yeah, I kind of guessed that might be the case. So what happens is NAD plus gets reduced, just like we saw FAD get reduced. And so again, we're focused on this primary place here, this nitrogen with the positive charge on it. And this will react with this double bond here will break. It will react with 1 proton and 2 electrons. And this double bond will break and you will get another hydrogen added over here. And so now this is NADH, because we've added 1 hydrogen to it, is the reduced form of NAD+. So instead of 2 hydrogens, it only requires 1 in order to do that. A common reaction that occurs with NAD+ is that it will oxidize a secondary alcohol to a ketone. So here we have a secondary alcohol. If you remember secondary alcohols, it is a carbon that has an alcohol on it, but it also connected to 2 R groups. It would be a tertiary alcohol if this was an R group here, but it's not, it's a hydrogen. So we will lose both of these hydrogens off of this secondary alcohol in this reaction, and you will get a ketone. This carbon will form a double bond here with that oxygen. And then you now have 2 free protons. One will be free and clear, but one of them will reduce that NAD+. So this is a common reaction with this. You'll see why this becomes very important. momentarily. Coenzyme A, which is also sometimes referred to as coenzyme ASH, uh, looks like this. Again, I'm not going to ask you to memorize the chemical structure of this. It's just the principles that are important. But again, we have this adenine base with a phosphorylated ribose. We see here that there's a phosphate group on this third carbon here. And then we do have two phosphates attached to this fifth carbon up here. And then we also have pantothenic acid, which is vitamin B5. So here's another vitamin B that is involved in this energy production in the cell. You're getting the hint I'm dropping here about vitamin B being very important. Might be a good test question when that comes around. But then it also has a thiol. So remember, we studied thiols. It's where instead of an OH, it's an SH. So this is 2-amino ethane thiol, because you do have an amino group here that's attached to this carbonyl group in the vitamin B5. And it is ethane because there's two carbons here. And then it's a thiol because it's a sulfur hydrogen over here. And this is the important site in this reaction. And what happens is this SH group will react to take the acetyl portion off and transfer that, gets transferred through this SH group. And this is why it's frequently also referred to as CoA-SH, because it's pointing out that this is the site where the acetyl group gets attached. So it's going to take this acetyl group of acetyl CoA and bond with it. So there are these three metabolic functions that occur in the body. As we've already seen, ATP can become ADP, which can become AMP, tri, di, and mono, but it can also go back the other way. And these are places of storage of energy through the transfer of phosphate groups. We also have these redox reactions that occur where FAD becomes FADH2. That's a reduction, but it can also go back the other way. And NAD plus can be reduced to NADH and can also go back the other way. So these are redox reactions. And then we have this transfer of this acetyl group to the CoA and back. So these are the three main metabolic functions that happen inside the body. Where am I at here? I think we'll go a couple more minutes and then take a break because I haven't given you a break yet. It's 7:15. We'll probably get done early again tonight. I know you're heartbroken, but so why are these phosphate compounds high energy? Like we said, it's because that they have highly reactive strained bonds. And these strained bonds get denoted with a tilde rather than a hyphen to show that they are in a strained state. It takes very little energy to break these bonds in a hydrolysis reaction, and therefore you get a net energy gain in the process. Strained bonds, the reason they're strained is because of the highly electronegative aspect of it. So in the phosphate, you got all those oxygens on there, or you have highly charged atoms will cause these strained bonds. And the free energy of these things are typically greater than 6 kilocals per mole. So here's some examples. We have an enol phosphate. So enol is because you've got a double bond here, but then you've got a hydroxy group that's been replaced with a phosphate. So it gets called an enol phosphate. And this is the strain bond. You've got all these electronegative oxygens out here, as well as this one here. This is a highly strained bond and will break very easily. And you can see the free energy is like 14.8. And so that's quite a bit of free energy. Acyl phosphates is where you have a carbon with a carbonyl group here, and then also the oxygen attached to the phosphate. This too is a strain bond, again, because of all the oxygens. It's a little bit lower free energy than enol phosphates, but still quite a bit. Guanidine phosphates. Again, you have a nitrogen here in these cases with a strain bond with this phosphate. And again, this will break off and produce more energy. But you can see it's now getting a little bit lower. Triphosphates, like we've already talked about, you got some R chain here with 3 phosphate groups on it. 2 of these bonds are very strained. And so this is where you can get ATP cleaving both of these off, giving you AMP plus 2 free inorganic phosphates. That's what this little I means, by the way, is stands for the fact that it's an inorganic phosphate. And then, or ATP can lose just one of these and become ADP with only one inorganic phosphate. And you see these free energies are still pretty good, but they're not as high as it's about half of an enol phosphate. But this is the principal engine for the energy production in the body. Diphosphates, like adenosine diphosphate, again, you're going to drop off one and become AMP and have a free phosphate ion. There are sugar phosphates, but this doesn't really have a strained bond in there. It's not as strained as these other ones that we've looked at. And you can see that in the much lower free energies of those bonds in here. Okay, why don't we take a break here? Why don't we do a 10-minute break and come back at 7:30? You guys are still seeing my screen, right? Okay, so this is an overview of what happens when you take in food and produce energy in your body. So you ingest food, and they could be fats, carbohydrates, or proteins. These fats are broken down into fatty acids and glycerols. Again, we've studied all of this before. Carbohydrates are broken down into glucose and other sugars, as we've studied previously. And proteins are broken up into their amino acid components. All of these can contribute to the formation of this acetyl coenzyme A that then gets entered into the citric acid cycle. The citric acid cycle will produce these reduced forms of NADH and FADH2, which will then be used in the production of ATP, which are these energy storing uh, components in the body. So we're going to go all the way through this. Stage one is where the digestion starts to break those down into smaller molecules. Stage two is where those small molecules from the digestion are degraded even smaller and produce these acetyl CoAs. Stage three is where the acetyl CoA is oxidized and it produces these reduced coenzymes of NADH and FADH2. And then stage four is where these two reduced forms facilitate the production of ATP. We're going to walk through all of this. So stage one, digestion, it actually begins in the mouth. Your saliva contains starch digesting enzymes. And so any starches that you're consuming start to get broken down immediately before it even leaves your mouth. Your stomach has gastric juices, which will contribute to the breakdown of that food. Your small intestine, as it moves from your stomach into your small intestine, it contains digestive enzymes and bile salts. Remember us talking about bile salts? These will help emulsify these breakdown products and bring it into your bloodstream. The end product of all of this is that you end up with glucose and other monosaccharides from the carbs, amino acids from proteins, fatty acids and glycerol from the fats and oils that you consume. And all of these get transported across that intestinal membrane into your bloodstream and get sent to the various cells in your body to be used as needed. Stage two is this acetyl group formation. This reaction occurs in the cytosol of the mitochondria. And this is where the small molecules get oxidized. And the primary products of this are that acetyl CoA that we've been talking about, as well as this reduced form of NADH from NAD+. Stage three is what's called the citric acid cycle, which I'm sure most of you have had before. It is also called the Krebs cycle after the scientist who discovered it. This occurs within the mitochondria. The acetyl groups get oxidized to produce CO2 and energy. Some of that energy is lost as heat, but some of that energy is transferred to stage four via the NADH and the FADH2. And your carbon dioxide that's produced in this citric acid cycle gets exhaled from your body. And this, by the way, is the principal cause of weight loss, believe it or not. It is you exhaling the carbon from your carbon dioxide that causes weight loss. I learned some years ago that this is the case. You will lose about 4 to 5 pounds overnight by breathing out carbon dioxide. That's actually how you lose weight. So if you take in fewer calories than you burn throughout the day, then you will lose weight. If you eat more calories than you burn throughout the day, you will gain weight. But overnight, you will lose about 4 to 5 pounds just due to the exhalation of carbon dioxide. And I've tested this. You could do it as well. Weigh yourself before you go to bed, weigh yourself when you get up in the morning, and you will be down about 4 to 5 pounds. And I just found that really surprising. I never knew that it comes from the carbon dioxide leaving your body. That's a lot of carbon you're breathing out. And then stage 4 is this ATP production. The NADH and the FADH2 will supply these hydrogen ions and the electrons that are needed to form ATP. And ATP is the primary energy that is carried in the metabolic pathways. The oxygen that you inhale, a good chunk of it is converted to water in this process. And ATP is formed through what is called oxidative phosphorylation, which all that means is that through an oxidation reaction, you're adding another phosphate group to that ADP to form ATP. And we'll see this in a moment. So again, just back to this high level view, all of your food ends up contributing to acetyl CoA. This enters the citric acid cycle. This will run around like this over and over again. So this is that cyclic metabolic pathway that we were talking about. But it will kick off these reduced forms, which will then contribute to the production of ATP in the body. Stages 3 and 4, these two stages here, are the same for all types of food, carbs, fats, and proteins. And the reason why is that they all basically end up producing this acetyl CoA. Stages 1 and 2 will differ depending on the types of food that you're bringing in because they break down differently. And this is what we'll be studying in the coming next couple of weeks. So the citric acid or Krebs cycle. It is named the Krebs cycle after Hans Krebs. He was born in Germany, served in the German army during World War I. However, when the Nazis took over Germany, his mother was Jewish and he was fired from his job and ended up leaving and going to England. There was some professors in England that thankfully were impressed with his work, and so they lobbied to hire him and get him out of Germany, thereby saving his life. A lot of scientists went through that during those times. His name is Hans Krebs. He's the one that discovered this as well as some other metabolic processes in the body. So again, that acetyl group gets oxidized to carbon dioxide, and you get these reduced forms of FAD and NAD+. It is called the citric acid cycle because one of the intermediate products in this cycle is citrate, which is a form of citric acid. So this is citrate. It is the anion form of citric acid. Citric acid would have a hydrogen on each one of these. Citrate is missing the 3 hydrogens and therefore has 3 negative charges on it. But this is why they call it the citric acid cycle. And it looks like this, which is a confusing mess. Again, I'm not going to ask you guys to memorize this or anything of that nature, but again, it's understanding the overall principles of the whole thing. And you may already have some of this memorized from other classes anyway. Who knows? So here is that acetyl CoA coming into this cycle, and it gets converted to that CoASH and also produces citrate. So this is why the very first intermediate species created is this citrate and why we get the name citric acid cycle. I told my wife the first time I taught this class that basically our bodies produce citric acid, so we're basically all like lemons with emotions because we're all just citric acid fruit. Um, this citrate reacts with a enzyme and will lose water and then gain water. We'll look at this in a minute as to why. And it forms isocitrate. This isocitrate then goes through the gets oxidized as this NAD plus reduces itself. So here we have this NAD plus becoming this NAD H and a free proton. And this forms alpha ketoglutarate. This then reacts with um, an enzyme again, and this produces carbon dioxide, a NADH from NAD plus, and also succinyl CoA. And then this will reduce, this is a phosphorylation reaction where we have GDP adds an inorganic phosphate and becomes GTP. And we also form this CoASH again. And it also produces a compound called succinate. And it's kind of small, but you can see these chemicals here on each one of these stages. In the book, it's probably much larger. You can look at it. This succinate then goes through an oxidation reaction where the FAD gets reduced to FADH2 and it produces fumarate. Fumarate then gets water added to it, so it's a hydration and forms malate. Malate goes through an oxidation reaction, and we have a NAD plus getting reduced to NADH, and this forms oxaloacetate. But oxaloacetate will react to form this acetyl CoA again and continue this process around and around and around. So this is a case where we've got A to B to C to D to E to F, G, H, I, and then back to A again. So this is a cyclical process. But we're getting a net reaction out of this. I can show you. So we have acetyl CoA. This is the net reaction of this whole cycle. Acetyl CoA comes in and it reacts with 3 NAD pluses, 1 FAD, 1 GDP, and an inorganic phosphate, and 2 moles of water. And it produces 2 moles of carbon dioxide. It forms a CoASH, 3 moles of NADH, this reduced form of NAD plus, 2 free protons, and the reduced form of FAD, FADH2. And we also get this GTP. This is the triphosphate created by this inorganic phosphate adding to the diphosphate. So this is the overall net reaction of this. So we're just going to go through each of these steps. So we have this oxaloacetate, which is the end product here of this whole cycle, reacts with this acetyl CoA. This is this acetyl group here on there. And it requires a citrate synthase, which will produce what's called a citral CoA, which then gets hydrolyzed and you get this citrate anion from it. And then you get the coenzyme ASH as a product as well as a free proton. Again, you guys don't have to memorize all of this stuff in that way, but again, just sort of understanding the overall process. So In step 2, we get an isocitrate forming. So we have our citrate from this previous reaction. So here we have our citrate as a product. This now becomes a reactant with an enzyme, and it will lose its water. So it loses an OH group and an H group from the citrate, and you get a double bond forming here. And then it picks up the water again using the same enzyme, and you end up with the water back on here. Why, do you ask? Well, what it does is it flips these. You see the OH group is on this central carbon, and now the OH group is on this second carbon from the bottom. It's basically flipped these around. And that's because we need this isocitrate in order to move on to the next reaction. So it loses its water and then gains the water again, just in a different configuration. Step 3, it takes that isocitrate from the previous reaction, and it uses a isocitrate dehydrogenase to reduce this NAD plus to NADH. We're going to lose, it's a dehydrogenase, so we're going to lose 2 protons off of there. One of the protons will reduce that NAD plus, and we will have a free proton as a result of that. And this will form oxalosuccinate. And you'll end up with this carbonyl group here because we've pulled off these 2 hydrogens. So you get a double bond forming there between this carbon and oxygen in this. This will then react with this free proton here, and you will cleave off the carbon and the 2 oxygens as CO2. So that's our weight loss molecule that's coming off and gets exhaled from the body. And the H attaches into there, and this is alpha ketoglutarate. The alpha ketoglutarate then reacts with a NAD plus, a free proton, and this coenzyme ASH with a alpha ketoglutarate dehydrogenase. So again, it's going to be pulling hydrogens off. And we have here this anion end gets the this whole anion comes off. We get this COAS part attaches into this carbon, and the hydrogen from this gets added to the NAD plus, thereby reducing that NAD plus to NADH. We've got more CO2 as a byproduct that gets exhaled off, and we get another free proton. So succinyl CoA is our product. That then gets used in step 5, where it is involved in the reaction that takes guanosine diphosphate, reacts it with a free inorganic phosphate, and produces guanosine triphosphate in this reaction and reforms our COASH. This succinate that forms as well then gets used in the next step, step 6, where it reacts with a FAD, again with another enzyme that's in the body, and this produces what is called fumarate. So here we've lost, it's a dehydrogenase again, So one of the hydrogens off of each of these carbons is pulled off. We form a double bond, and we end up with FAD-H2. I know I'm going through this pretty quickly, but if you have questions, just pipe up and ask. The seventh step then is it takes that fumarate from the previous reaction and it gets hydrolyzed. We're adding water onto this. It's going to break that double bond. It's going to get hydrated, I'm sorry. We're going to add that double bond. We're going to break that double bond and add the water to the carbons. And so you get a hydroxyl group attached and a hydrogen group attached. And this is L-malate. And then finally, the last step is the oxaloacetate gets regenerated again. So the malate reacts with a NAD plus and another enzyme, and you lose the hydrogens again. You get a double bond forming here, and you're back to oxaloacetate, and you have NADH plus a free proton. And so if you add all that up, we get back to this net reaction where you end up introducing acetyl CoA, and then you end up getting a high energy GTP formed, as well as these reduced species, which will be used to form ATP, as we'll see in a minute. So all of these reactions happen in that mitochondrial matrix, the inside of that curly space that we saw in the diagram, except the sixth step, which is in the inner membrane. The fuel of this whole reaction is this acetyl CoA, which comes from the carbs, the fats, and the proteins that we consume. Do I have the diagram? No, I guess I don't. But remember, all of those get broken down and produce this acetyl CoA. That's the fuel for the citric acid cycle. There are 4 redox reactions. There's 3 involving NAD plus and 1 involving FAD. And the NAD plus forms these carbonyl groups, and the FAD performs these alkene groups. The NADH and the FADH2 are what carry these hydrogens and electrons that will form the ATP from ADP, and we'll see that in a moment. And as I've been saying, there are 4 vitamin Bs are involved in this whole process. There's riboflavin, nicotinamide, pantothenic acid, and thiamine are all vitamin Bs that are involved in this whole process. And you do get this one high energy GTP formed as well, which your body can then use for energy later. So I could see possible, like I said, I'm not going to expect you to draw this out, or I'm not going to ask you to draw oxaloacetate or anything like that. What I might ask you is like, what is the fuel of the whole overall reaction? Or, you know, I might ask you like, you know, um, I'm really talking off the top of my head here, so I'm trying to think of things like, you know, what 2 species get reduced in this overall reaction might be a type of a question I might ask. That kind of a thing. I'm not going to ask you guys to memorize all of this.
Thank you. That was my question. If we had to memorize all those steps.
Yeah, go ahead. If it was April Fool's Day, I'd tell you guys that. Do you know I didn't play any April Fool's joke on Friday? Um, just didn't even think about it. I got very busy. But I think again, it's understanding. So another question might be like, you know, how important are enzymes in this overall process? So Erin says she's had to memorize this so many times. Um, understanding that enzymes are used in almost every single one of these steps in here uh, is important. Um, I could potentially, I don't think I have this in the exam, but I haven't looked over the exam, but I could potentially ask like, uh, what is getting reduced in this? I could show you this reaction and say, what is getting reduced in this? And the answer would be NADH. The NAD plus is getting reduced to NADH, something like that. I don't know, but don't have to memorize it. So you had to memorize these or just sort of the steps, Aaron?
So I had to memorize this entire thing for one of my physiology classes in my undergrad. And
Wow.
then we basically had to draw out the entire thing with all the enzymes and reactions and stuff.
Wow.
Yeah.
Yeah, I'm not going to be that mean to you guys. It was
Thank you.
actually a little hard picking out practice problems because the book also kind of expects you to do that. And I'm like, no, I'm not going to do that to you guys. I'm more interested in you just understanding the overall process of it all. Okay, this process could run amok being cyclical, except we do have two rate limiting steps and or what's called regulation that regulates this overall reaction. There's one at step one because high levels of ATP in the cell inhibits the citrate synthase, and so therefore this first reaction will slow down. So we have this citrate synthase, which is this enzyme that helps you take this oxaloacetate and the acetyl CoA and form that citrate, and so it inhibits this. So your cell, it's forming those NAD, um, it's forming these NADHs and FADH2s, which will then go on to produce ATP, as I mentioned here, and we'll see this in a moment. So as the concentration of ATP rises, it will then quell the reactivity of this, and so it will regulate and tone down the that whole overall process. When the ATP drops because it's being consumed for the needed energy in the body, then you're going to have more ADP because this ATP is going to break down to ADP, and then this will cause that citrate synthase to become more reactive again. Does that make sense? And also the third step in NADH inhibits the isocitrate dehydrogenase, but it gets activated by ADP. So again, as NADH is produced, it will slow down that third step, and as it is consumed, it will allow it to reactivate and speed up again. Oh, there's my overall diagram. Yeah, so this first step and this third step get regulated. in that way. So that keeps this overall process from just going crazy. Don't know why I have that there. I guess just to say that now we're going to look at the formation of the ATP. And if you remember, those sites are on the inside of this cristae, this membrane wall inside of the matrix. And so we're going to look at how this ATP gets synthesized. So what happens here is we have the inner membrane space is on the top in this diagram. So that's this space that's in between the outer membrane and the matrix itself. So it's this area in here, the intermembrane space. And then at the bottom here, we have the matrix. So that's this space inside of this inner part of the mitochondrion. Oops. And that, remember, you're going to have a bilipid layer here that forms that membrane. So we have the polar heads pointing at each other with the nonpolar tails on the outside. You can see it in my picture behind me there as well. And so we have various species here that uh, either breach that bilipid layer or are attached to one side or another. And again, I'm not going to make you name or memorize what these are. These are just called protein complexes 1, 2, 3, and 4. And then we also have this ATP synthase, which does go across that membrane layer. And what all of these do is it helps the body produce an equilibrium of hydronium ions, protons. So what happens here is in the matrix, remember we're producing NADH and FADH2 in that citric acid cycle. And these will get oxidized back to NAD plus and FAD and produce free electrons in the process. And these will feed what is called the electron transport chain. It's abbreviated ETC, etc., I guess. And this is a series of redox reactions which will activate these pumps in the membrane to move protons through from the matrix out into the intermembrane space. So this first one gets activated as free electrons flow into it. This will begin working and it will pump these protons that are produced in the matrix out into the intermembrane space. Those electrons will then move from this first protein complex to a species that's called uh, coenzyme Q. And I think I have a diagram here of it. You can see this long, crazy chain thing. And it's got like all this stuff we've been studying all along, right? We've got this huge chain with lots of double bonds in it. And it's got a ring structure with carbonyl groups on it, as well as these are ether groups. We've got an ROR and an ROR here in this structure as well. And what this will do is it will take the electrons from that first protein complex. It will also receive electrons from this second protein complex. So this second protein complex does not go all the way across. So it will not pump protons through like the first one does, but it will accept electrons and it will pass them on to this enzyme as well. And then this enzyme will take the electrons from both of these 1 and 2 complexes and pass it to the third one. This third one then gets energized and it will pump protons across the membrane out into the inner membrane space. And then these electrons will then move from the third one to another protein that sets on this side. So it's setting on the outside of this inner membrane space. And this is called cytochrome C, which I have a diagram here. And this is abbreviated cytC. And you can see it's a protein. I wanted to put this picture up just so you could see here. We've got this protein that's got a alpha helix structure here and here. But we also have some unstructured parts of that protein as well. But it also has a heme. Remember, a heme is that structure that has an iron that sets in the middle that will react with the oxygen. And so this receives electrons from all three of these now, and it will then use that, pass it on to the fourth protein complex, which will then activate and pump even more protons through from the matrix out into the inner membrane space. And then these electrons will leave this complex and come back into the matrix again. And your oxygen in here will react with 4 protons and 4 electrons and produce water. That was a mouthful. And so remember I said earlier on that the oxygen ends up producing water in our bodies. This is the reaction that occurs. So this whole process here is the electron transport chain. We're going from here into here, from here into here, both over to here, both up to here, then into here, and finally dropping back out. All of these, then these 3 are putting protons out into this inner membrane space, and this now is creating a gradient across here. We're going to have fewer protons inside the matrix, and we're going to have more protons in the intermembrane space. And when this concentration gets high enough, it activates this ATP synthase, and it will take the protons in, and the protons act as a catalyst for this synthase to take an ADP and react it with a free inorganic phosphate to form ATP. And this, therefore, stores energy. Everybody's having uh, connection problems tonight. Everybody keeps dropping in and out. But since this proton is just a catalyst, somebody's giving us feedback. Since this is just a catalyst, we're now then going to increase the proton concentration inside the matrix. And then as this concentration gets higher, and as our body produces more NADH and FADH2, this is going to cause this to start up again. So you get this cyclical structure process of all this. Does that make sense? So the gradient out here is going to become higher. This will push the synthesis of ATP, but then the proton gradient will build inside of here, and then that will push it to go back through this way as well. But if you're not eating food, then you're not getting more NADH and FADH2 from that citric acid cycle, and therefore this process will shut down eventually. Uh, but as long as you're bringing in and producing more of this, then you're going to have plenty of free electrons to keep this cycle running over and over again. Any questions there? Okay. There's my little chemicals again. Okay, so from one acetyl CoA, remember that's what gets fed into the citric acid cycle. 3 NADHs will make 7.5 ATPs. 1 FADH will make 1.5 ATPs. And one, that guanosine triphosphate will make 1 ATP for a total of 10 ATPs. So we get 10 high energy, this is the energy is stored in this ATP molecule from just one acetyl CoA. So we've magnified this tenfold through this whole process. These ATPs then are available when our body needs energy, where we do that anabolic process of building things. Then it's going to strip off one of these phosphates, get free energy from that, and you're going to end up back to ADP again. And that free energy will then allow our bodies to build the chemical structures that we need. Glucose, which has 6 carbons, produces about 30 to 32 ADPs. Stearic acid, which is C20, produces about 100 ATPs. So you get a lot more energy out of this one because it's got so many more carbons in it. So it's going to produce more acetyl CoA basically. Uh, let's see here. Any questions there at all? So I just wanted to flip back. Oh, I have it right here. Cool. I didn't remember this. So yeah, so what this is, this whole process then gives us those ATPs that can be used in that whole process again. So we're breaking down the food, and this is going to produce these ATPs. That's that whole cycle that gives us that. Do I have the diagram? Okay. This citric acid cycle is going to give us these NADHs and FADH2s, which are going to feed that electron transport chain, and that will produce these ATPs. So we're going from ADP to ATP in that whole process. But then we need to work our muscles, so we need energy for that. We need to move nutrients throughout the body. We need to synthesize compounds in our body. This is an anabolic process, and so it's going to cleave off a phosphate to get energy. Remember that strained bond? So that'll pop off to give us ADP again, and then that will allow us to build proteins and so forth in our bodies, and so that's where the energy is coming from, is these stored up adenosine triphosphates. So that's the kind of overall stuff that I want you guys to focus on, then rather than memorizing chemical reactions or whatever. So you might get a question on the test like, you know, what compound is significant in an anabolic uh, metabolism? That would be the ATP getting consumed to produce the energy to build again. or something like like that. So that's the kind of questions you're going to get, not all those chemical diagrams and everything. Okay, 90% of the oxygen that we take into our bodies, that we breathe in, gets used in the synthesis of ATP. 10% of the oxygen we take in forms what are called reactive oxygen species, ROS for short. And these are things like hydrogen peroxide. Yes, our bodies have hydrogen peroxide in them. Superoxide, which is just O2 minus. Hydroxyl radicals. This is an OH group that's a free radical of OH. And these all aid in their very highly oxidizing chemicals. So they will help in the destruction of bacteria and viruses, as we've kind of talked about before. But these can run amok as well. And so what they get regulated by is vitamin C. Vitamin C is that antioxidant that keeps these things from building up too much in the body and starting to destroy things that you don't want destroyed inside your cells. So vitamin C is very important in regulating that. So again, we just have this overall thing. We're going to eat food. The food consists of fats, carbohydrates, and proteins. These are all going to break down and ultimately give us acetyl CoA, which will then enter into the citric acid cycle. The citric acid cycle is going to run, and it's going to have a byproduct of CO2 that we exhale and lose weight. It's going to have two main products of NADH and FADH2, which will feed into the electron transport chain, which allows our cells to activate that ATP synthase and produce ATP, thereby storing energy in our bodies for when our cells need the energy. Any questions there at all with any of that? Everybody good? Okay, well, the last thing I have is I found this last year when I was preparing for the class. There's actually a really cool animation video I found that kind of explains this last part here, this whole process of the ETC. So we'll watch that. And again, this is in the slides if you want to watch it again later. So it's kind of got like a little animated thing. Here you see these complex 1, 2, 3, and 4. And then here is that ATP synthase. And we're going to see it all in action.
The human body contain membrane-bound organelles.
Hang on, I got to turn the volume up.
Mitochondria are double-membraned organelles that harness.
So you guys hearing this okay?
Most of the energy that cells need to grow and reproduce. Nearly all of this energy comes from reactions that take place at the inner mitochondrial membrane.
So there's that bilipid layer.
One of the key roles of this membrane is to act as a barrier to positively charged particles called protons, thus allowing a concentration gradient to be maintained where the intermembrane space has far more protons than the matrix. The membrane also contains a large protein complex called the F1F0 ATP synthase, which uses the proton gradient to drive the synthesis of ATP molecules. These ATP molecules ultimately provide the energy for most of the cell's reactions. Just as man-made power plants produce electrical energy by using the flow of wind, water, or steam to rotate a turbine, the synthase makes ATP by using proton flow from one side of the inner membrane to the other to rotate protein subunits. If there is no proton gradient, synthase subunits stop rotating, and the cell can quickly become starved of energy and die. Therefore, the protein complexes and small molecules that establish this gradient and maintain it play an essential role in the life of the cell. At the heart of this system are 4 protein complexes, numbered 1 through 4. Complexes 1, 3, and 4 directly pump protons from the matrix into the intermembrane space. Complex 2 does not directly pump protons, but it does promote proton pumping in complexes 3 and 4. Proton pumping requires energy, and the 4 protein complexes get this energy by transferring electrons through a series of coupled reactions. This linked process of electron transport is why the 4 complexes are collectively referred to as the electron transport chain. Let's focus on complex 1. A byproduct of sugar metabolism called NADH deposits 2 high-energy electrons in complex 1, where they are passed along a chain of redox centers. Redox centers are clusters of atoms that have different affinities for electrons based on their unique atomic configurations. Let's closely consider a pair of redox centers to reveal 2 reasons why an electron moves from the top redox center to the bottom. First, the bottom redox center has higher affinity than the top one. Second, the distance between these adjacent redox centers is ideal for an electron jump to occur, which explains why electrons typically don't bypass the bottom redox center. A small amount of energy is released each time an electron is passed between redox centers. Complex 1 harnesses this energy across all the redox centers and uses it to pump protons.
So those yellow balls are the protons moving through.
The last redox center in complex 1 donates 2 electrons to a coenzyme Q molecule. Complex 2 is similar to complex 1 in 2 important ways. First, high-energy electrons also enter complex 2 via a byproduct of sugar metabolism, although here the molecule is FADH2. Second, complex 2 also transfers electrons between several redox centers before donating them to coenzyme Q. One major difference, however, is that complex 2 does not use the energy liberated to pump protons. Coenzyme Q molecules from complexes 1 and 2 donate their electrons to complex 3. One electron is recyclable and can re-enter complex 3 later, but the other passes through 2 redox centers before reaching cytochrome C. Cytochrome C carries the electron to complex 4. The electron transport chain ends in complex 4, where a series of reactions involving 4 electrons converts a molecule of oxygen to 2 molecules of water. The proton gradient is strengthened because 4 protons from the matrix are incorporated into water molecules, and another 4 are pumped into the intermembrane space. In the absence of oxygen, the electron transfer comes to a halt, meaning that ATP synthesis also stops. Indeed, the reason we breathe oxygen is so that it can serve as the final electron acceptor at the end of the electron transport chain. In this animation, we have explored each protein complex in isolation, but in reality they are very densely packed. Together, they effectively make the entire surface of the inner mitochondrial membrane a giant cellular power plant.
I just still continue to be fascinated by this. Our bodies are amazing. Just
Professor, can you clarify, is this a process that then happens in all of our cells or just specific cells?
it would be most of our cells that I know of have mitochondria in it, and that's where this is happening at. So might there be cells that don't do this? None that I'm aware of, but I'm not a biologist and I've not heavily studied that. So I think it's primarily all of our cells. Does anybody else know the answer to that? I can't imagine a cell not having this in it, but then again, like I said, I'm not that knowledgeable about it. Isn't that just like absolutely amazing? So these little molecules act like, rotating like wind turbines and things and stuff. It just absolutely amazes me. Like I told you guys before I taught this course, I knew none of this stuff. And I just found it absolutely amazing. All my background was in industrial analytical chemistry, not biochem. It was all the pre-med kids had to take biochem, but I didn't have to take it. And I just found this absolutely fascinating. I still am, but just watching that again, it's just like really just incredible what our bodies do. Okay. Do have some problems here again for you to look at. And, we will review chapter 12 next week. Aaron says, I think all cells do since mitochondria are in all animal cells, right? I believe so. I mean, I'm reaching way back to 9th grade biology, and that was many years ago, guys. Way more than I'd like to admit, but I'm pretty sure there's mitochondria in everything. Because your cell wouldn't be able to produce energy if it didn't have it. So the cells would die. I think you're right on that, but I'd have to look into it to know for sure. Now, what probably doesn't have this, again, I say probably, so don't count me on this, but things like viruses and stuff, they hijack the body's processes to do things. They can't do it on their own. And so they may not have mitochondria to do this. But I'm not sure of that. But anyway, so next week we will review we will review the exam from this weekend. We will review chapter 12 and then do 13 and maybe part of 14 we'll see as we're winding down. So, any questions, comments, anything before we call it a night? From anyone? Well, if not, everybody